Hi, I'm Sairah Malkin. I'm an assistant professor at Horn Point Laboratory, and we're standing right now in front of the Choptank River, part of a tributary to Chesapeake Bay. So I'm going to talk about microbial ecology that I'm studying in Chesapeake Bay. Microbial ecology is the study of microorganisms, or short form microbes, in the environment, and their interactions with each other and with the environment. Microorganisms include both the phytoplankton that we study in the bay, and phytoplankton in the oceans are responsible for about half of the oxygen in the atmosphere. Microbes are also important for degrading organic material, so returning carbon and nutrients back to the soil. In fact, microbes are responsible for building the soil on the whole planet. So when we think of microbes, although it is important to consider the pathogenic ones, it's also important to consider that most are harmless, and in fact, microbial ecology is essential for our weather, our soil, our atmosphere, the functioning of our aquatic systems, our personal health. They're essential to all of life on the planet. So what I'm going to talk about then are a couple different bacteria that we find in aquatic environments, and particularly in Chesapeake Bay. I study sulfide in the environment. And you're actually familiar with the compound sulfide. It's produced under conditions of no oxygen. It is produced and can be found in waterlogged soil, in sediments, and even in the deepest parts of the bay when it goes anoxic, when oxygen is removed. And you're probably familiar with it if you've ever gone walking through a marsh. You get that smell of rotten eggs. In part, what you're doing is releasing sulfide from that waterlogged soil. So that's really what sulfide is. And it's highly toxic. It's toxic to plant roots. So if roots are growing down in waterlogged soil, then sulfide can diffuse into their root system. Sulfide is also toxic to all animals. So if there's a massive, for example, algal bloom that rots on the seashore, then that's also producing a lot of sulfide that can be harmful to pets. So we're fairly familiar with the toxicity of sulfide. But there's some organisms that can actually get energy from that sulfide. So I study those bacteria that feed on sulfide and thereby remove it from the environment, detoxify those environments. So I'm going to talk about two specific ones that are found in Chesapeake Bay. One of them is called Beggiatoa. That name doesn't really matter. I'm going to refer to it as the scuba diver. It was first discovered in the late 1800s. And the reason it was described so early is because it's actually one of the largest bacteria that we know of. It's very long. It grows cell to cell to cell to cell, all together in a long line as a filament, like a human hair. And it grows to about an inch or more long. And it's really thick. So not only is it very long for a bacterium, but it's also really thick, really fat. And the reason it's so big is because it can fill itself with what I'm going to call scuba tanks. It's scuba tank. They're technically called vacuoles. But what it does is it lives in the sediment, and it moves up to the sediment surface where it fills its scuba tanks, its vacuoles. Then it dives down into the sediment, and it respires from its scuba tanks, and it consumes sulfide in doing that. So it moves up and down in the sediment, and it's very efficient at removing sulfide. I'm going to contrast that with another bacteria that's only been discovered in the past 5 or so years. And it's called cable bacteria. And it's called that because it effectively functions as an electric cable. And what it does is it plugs itself in to the surface of the sediment and grows downward in the sediment. And it takes what electricity is, it's the flow of electrons. So it takes electrons from deeper in the sediment, and they're conducted across the length of this bacteria up to the sediment surface where they're discharged to oxygen. This very interesting thing that we found, that even after these electric cables, the cable bacteria die off, the sediment still keeps the sulfide trapped for weeks even after the cable bacteria have died. That's not necessarily true of the scuba divers. So there's some really strong differences between the two communities. So why does this matter? Well, one of the things we're really interested in is why some of these bacteria are found in some places and these other bacteria are found in other places. So there's very different effects on the ecosystem. They both have very different influences on the sulfide that's in the sediment. And we need to better understand why we get sulfide in the environment, we need to understand the controls on the different microbial communities that are there.